Welcome to FYI, the For-Your Innovation podcast. This show offers an intellectual discussion on technologically enabled disruption, because investing in innovation starts with understanding it. To learn more, visit ark-invest.com.
ARK Invest is a registered investment advisor focused on investing in disruptive innovation. This podcast is for informational purposes only and should not be relied upon as a basis for investment decisions. It does not constitute, either explicitly or implicitly, any provision of services or products by ARK. All statements made regarding companies or securities are strictly beliefs and points of view held by ARK or podcast guests, and are not endorsements or recommendations by ARK to buy, sell, or hold any security. Clients of ARK Investment Management may maintain positions in the securities discussed in this podcast.
Welcome to FYI, the For Your Innovation podcast. Today, we're going to be talking to Andrew and Seik from Verve Therapeutics. Andrew is the CSO and CMO, and Seik is the co-founder and CEO. So by way of short background, although it's hard to give a short background to these two, Andrew is a cardiologist and scientist. He worked at the Brigham and Women's Hospital. Andrew co-founded and was the chief scientific officer of Verve Therapeutics, also was the chief scientific officer of Cocoon Biotech and co-founder of Corner Therapeutics. Andrew's training is in internal medicine, which he did at the University of California, San Francisco, and then his cardiology training at the Brigham and Women's Hospital. And he completed his postdoctoral research training in drug discovery at MIT, and he holds an MD and a PhD from Columbia and an MS in mathematics from New York University and an AB in physics from Princeton. Sorry, that's a mouthful. Andrew, you let me know if I missed anything. Seik is also a cardiologist and scientist. He was the director of the Massachusetts General Hospital Center for Genomic Medicine, director of the Cardiovascular Disease Initiative at the Broad Institute, and a professor of medicine at Harvard Medical School. Seik holds a BA in history from the University of Pennsylvania, received his MD from Harvard Medical School, and completed his training in internal medicine and cardiology at MGH, and postdoctoral research training in human genetics at Framingham Heart Study and the Broad. So welcome to the podcast, and we're so happy to have you on.
Delighted to be on, Ali. Thank you so much.
Great. So you can let me know if I missed anything, but we could probably go on for just an hour just about your backgrounds. So to pivot a little bit, just a really short intro to what Verve is. It's a biotech company that's bringing new approaches for cardiovascular disease, and we're transforming the way we think about chronic management for these conditions. So including even gene editing single-course treatments. So really excited to discuss all that is gene editing and cardio. So again, sorry, that was all a mouthful, but just wanted to give good context. And now we can kind of dive in. So wanted to give you, Andrew and Seik, an opportunity, if I missed anything or if there's something you want to add in terms of background, how you got to this point.
Well, maybe I can start just to give you a sense of, why we are here in terms of trying to develop gene editing medicines for atherosclerotic cardiovascular disease. Both Andrew and I trained in cardiology. We care deeply about atherosclerotic cardiovascular disease, which is the leading cause of death in the world, so heart attack. And what we have learned over the years is that if one's LDL cholesterol, the bad cholesterol, is very low, lifelong, it's really hard to get a heart attack. And our company is founded on the idea that we can develop a one-and-done gene editing medicine that would lower LDL cholesterol dramatically and do so for essentially a lifetime, durably do so, and as a way to treat heart attack. And this was a concept that we came up with back in 2016. Took a couple of years to develop that idea. In 2018, we founded the company, and then over the last 4 years have really worked to develop proof of concept now in monkeys that this is going to work, that a one-time treatment, permanent lowering of LDL cholesterol as a way to treat heart attack. And so excited to share with you that story and the progress.
Great. Andrew, anything to add?
No, I often say, being the CSO at Verve is the perfect job for me because, I am a cardiologist at my heart. My background and experience is really in translating new therapies. And I think, as Sek said, the central challenge here, we knew that the biology was there. The central challenge was, could we make it work? And that's a problem that, it's the kind of problem I like to solve. So it's been a lot of fun working with Sek the last 3 years on tackling that big vision.
And Sek, I read an article that really actually moved me about a personal tragedy that you had with cardiovascular disease and your brother. So don't want to put you on the spot, but if appropriate and if you feel comfortable, maybe if you wouldn't mind sharing. I think you had already been a cardiologist at this time, but I can imagine it sort of further ignited your passion to find real potential cures and maybe even change the way that we're treating cardiovascular diseases in general.
Ali, the story you're referring to is that of my brother. We have a strong family history of coronary heart disease, of heart attack, premature heart attack, and my brother and I kind of knew that. We had come to this country back in 1980 as immigrants. My father was already here. We grew up in Pittsburgh. I was on staff as a cardiologist at Mass General Hospital. My brother living in Pittsburgh with his wife and my niece, and he'd gone for a run, came back, was dizzy, and ended up calling an ambulance, and then he passed out in the house and was resuscitated by the EMTs. But taken to the hospital, turned out it was a blood clot in one of the heart arteries. So he had just suffered a heart attack. That blood clot was treated, but he had actually suffered a fair amount of anoxic brain injury during the resuscitation, prolonged at home. Unfortunately, died about 10 days into the hospital course, and that tragedy. So he was 42. And I had been studying basically the genetic basis of heart attack for the previous 10 years or so. Really struck home that the problem that we were working on and the solution that we were looking to arrive at, it really is even that much more important that we tackle this. It really recommitted me to this issue, and not just the issue of diagnosis and understanding why somebody's at risk, but more importantly, the idea that we should develop a new approach, a new way to treat, to avert such tragedies. And so it was around that time, to be honest, that I kind of tried to think about not just the research we were doing, but how can we translate that to, as Andrew said, to new treatments. And it was right around that time also that the whole gene editing revolution was happening, right, in 2013, 14, with new discoveries, and it's really that confluence of events that got us over the subsequent 3 or 4 years to this idea of developing a treatment so that things like what happened to my brother don't happen.
Very sorry to hear that, Sek, but it's amazing what you've done sort of post that and to really try and help patients. So maybe if we can move to Verve specifically and talk about your sort of 2 programs that we know announced now, obviously PCSK9 as the first. Can we kind of talk about why you chose that as the first program and kind of the progress that we've seen so far?
Yeah, and just taking a step back, as I said, atherosclerotic cardiovascular disease, and this is basically blood LDL clogging the heart arteries, is the leading cause of death worldwide. And we know that, as I said, lowering the LDL cholesterol as low as possible for as long as possible is the major treatment to make sure you don't have another heart attack if you've already suffered one. And so, and there are some treatments out there available to lower LDL, but all of those treatments follow the same model, which is the chronic care model, the daily pills, the intermittent injections. And that model is broken. And what do I mean by that? Well, that model requires a bunch of things: adherence, access, and has lots of issues. And a very simple stat kind of will highlight this. So if you take 100 patients who suffer a heart attack today and ask a year from that time period, year from that heart attack, how many are still taking their cholesterol-lowering medications? How many are still on their cholesterol-lowering medicines? It's only about half. And so that's all really due to the chronic care model. And what we want to do is completely change that and say, what if we had something that, one-time treatment, dramatic and durable lowering of LDL? So that's really the concept. And so to actualize that concept, we had to say, what are the right targets? And then what's the right approach to do this one and done? The first target we've chosen is PCSK9. In fact, we had a basket of targets, 4 different targets we selected when the company started. And Ali, all those targets have the same profile. They are that there are humans walking around who naturally lack the gene, have the gene turned off, and have low LDL cholesterol and are protected from heart attack. So there's human genetic evidence. The targets also have human pharmacology evidence. So that means that there's another modality that's been developed against that target, and it's been shown to work in people. And the human genetics and human pharmacology is really the foundation for why we selected. And that's the reason we chose PCSK9. Then the next step is, how can we turn off this target permanently? And our target organ here for this gene is the liver. We want to turn off the PCSK9 gene in the liver. And the approach that we prioritized is base editing. And we got to that after evaluating both standard CRISPR-Cas9, which is basically cutting DNA to turn off a gene, and also base editing, which is like a molecular pencil and eraser to basically change a single letter to turn off a gene. We compared both those approaches in cells, mice, and monkeys. And then, as I said, I prioritized the base editing approach. And maybe I'll let Andrew walk you through the preclinical data we've generated for our first product, Verve 101, which is an adenine base editor and a guide RNA targeting that base editor to the PCSK9 gene packaged in a lipid nanoparticle. Andrew?
Yeah. So the first step is identifying, as Sek said, where in the PCSK9 gene you want to make a single base pair edit. And so we screened a large number of guide RNAs targeting PCSK9 to identify one that is unusually specific and is able to make a single base pair change at the end of exon 1 in PCSK9. One base pair change of A to G will, in that splice donor at the end of exon 1, will result in essentially no protein production of PCSK9. So we had identified those guides and then took them into mice and then into non-human primates. And what we found is that with a single IV infusion of Verve 101 in non-human primates, we can, with essentially 100% fidelity, reach almost every liver cell, every hepatocyte, and edit both copies of the PCSK9 gene, that A to G change, and turn off PCSK9 production. So we see 90% reductions in PCSK9 levels circulating in the blood just a week or two after we give Verve 101 in non-human primate. And those results now have been durable out in our earliest studies out to more than 15 months. We've done a large follow-up study with the actual clinical candidate Verve 101 and have durability data we presented last year out to 6 months. And that large durability study had 36 monkeys in it. It was a very robust study. And what we saw was very consistent reductions, high-level editing in the liver, about 70% in the whole liver sample, which reflects the vast majority of hepatocytes, and a 90% reduction in PCSK9. The result of that was that we could lower LDL cholesterol by 60 to 70% just 2 weeks after the one-time infusion, and it stayed down for the duration of the study so far.
What's remarkable is, this is like surgery without a scalpel. You're literally able to go in, make a single spelling change in every liver cell, and turn off a gene, a disease-causing gene. And what you have then is basically permanent lowering of LDL cholesterol after that one intervention.
There are other modalities, though, that can be used for PCSK9, right? So maybe it'd be helpful to talk about some of the other potential treatments that can be used. And of course, as you stressed and said, this is potentially a one and done. So that's kind of the biggest differentiator this potential therapy has. But just curious of how you think about the treatment paradigm for PCSK9 as it stands now.
Yeah, so there are other treatment modalities. There's a monoclonal antibody that targets a circulating PCSK9 that's injected typically twice a month. There's also a siRNA targeting PCSK9 that just got recently approved. And there's even other modalities, even an oral PCSK9 inhibitor that's being developed. All of these current and treatments in development against PCSK9 follow the same model that I mentioned earlier, this chronic care model, requiring daily pills or repeat injections. And that's the model, as I mentioned earlier, that's broken. And as evidenced by that stat that I mentioned, despite all that's available, people are not on their medicines about a year after their first heart attack. And there's a consequence to that, Ali, which is instead of having the LDL come down and stay down as it's supposed to, there's oscillation in the LDL in the patient's life course after their heart attack. And that lack of control over their life course leads to recurrent heart problems like a heart attack or a bypass surgery or even a stent or even a fatal event. And that's really what we're trying to overcome. That's the unmet need, really to convert the LDL care from that oscillation to just down and stays down. And that's really what our product will look to do.
Heterozygous familial hypercholesterolemia is a fairly common disorder, but it's a very morbid one. It's one with a high morbidity and mortality. So these are patients who have typically a mutation in the LDL receptor or one of a couple other genes that results in elevated LDL cholesterol from birth. So these patients are accumulating cholesterol in their arteries, all through their childhood and young adulthood, and then typically have heart attacks in their 30s, 40s, and 50s. So these are, we often think of kind of cholesterol as a common disorder, and maybe something that we can't do much about. But in fact, the consequence, as Sek said, is that these people actually suffer clinical events that have a huge cost to themselves and to their families. And so that's really where we think that these kind of therapies can intervene in these, initially at least, in these high-risk patient populations with a genetic disease where a one-time genetic therapy, a gene editing medicine, can provide a durable solution for them. And then with sufficient data in those patient populations, that's when we can really think about going into even broader indications. But it's important to know that first indication of heterozygous familial hypercholesterolemia is still a pretty common disorder. So about 1 million people here in the US have this genetic condition that predisposes them to heart attacks and stroke.
I get asked a lot about sort of what we think of as society changing and medicine changing. So more of an upstream approach versus a downstream approach. And I actually love the Verve example of how we can think about healthcare from the upstream approach. So, when we talk about your model as the 3 phases, so maybe that's something we can talk about, just how you see it sort of in a phase approach where, first we're tackling X group of people and then following to make it an upstream. This is what you're talking about, Andrew, right? So not focusing only on those people who necessarily are prone to having a heart attack, but really that this could potentially treat everyone.
Yeah, no, I think that's what's really very unique about LDL cholesterol. So if you've developed a heart attack, the number one thing to do to make sure you don't develop another one is to lower your LDL cholesterol as low as possible for as long as possible. Now, it turns out to prevent a first heart attack, the main thing to do is to keep your LDL as low as possible for as long as possible. So given those observations, we have a phased approach to our clinical development for our lead product. So Andrew mentioned the profile, which is one-time infusion, 60 minutes, let's say, intravenous infusion. You walk out and LDL goes down 60 to 70%, stays down. And we think this is going to be lifelong, and we can talk about durability maybe in a few minutes. But that's the profile. LDL comes down, stays down. Which patients are we going to apply this therapy to first? Well, the first group is heterozygous FH. This is a genetic disease. They have high LDL not because of their diet or lifestyle, but because of a DNA mutation. This is going to be a genetic treatment for their genetic disease. That's about 1 million people in the US, another 1 million in Europe, 31 million globally. Okay, that's the first group. And the second group would be anybody who's had a heart attack in the United States or globally. And that's, the umbrella term is atherosclerotic cardiovascular disease. That's about 22 million people in the United States, similar numbers in Europe, and then about a couple 100 million globally. So again, that group would stand to benefit from lifelong LDL lowering. And so that's the second patient population. And then ultimately, with a sufficient safety database and efficacy database in those first two groups, Ali, we could see this medicine being given to patients to prevent a first heart attack before they have their heart attack. And that, addressable market is literally most of the world. So there's a development, there's a stage development, a phase development from FH to ASCVD to prevention, all with successively larger addressable markets.
A question we often get asked, Ali, is would patients really sign up for a gene editing therapy for these more common disorders? And I think I think a few things have happened recently. One, certainly as we've been reaching out to patients and clinicians over the last year, we've seen tremendous enthusiasm. Patients are interested in this. Now we're talking mainly to patients who, surveys of patients with heterozygous FH. But I think what we're also seeing is that the idea of a lipid nanoparticle that carries mRNA in it being something that can be administered to millions of people or even billions of people now has become much more clear, in part because of COVID. Because the COVID vaccines now have really demonstrated the feasibility of manufacture, the feasibility of administering these to large numbers of people, and that people, that a new technology can have a dramatic impact on medicine.
I think one of the exciting things also is that you guys are technology agnostic. So when you say that you found, that base editing for this particular indication was, the right technology to use, it doesn't mean that you'll be using base editing or any other one technology for the rest of your pipeline. So can you kind of talk about how you see the pipeline emerging in terms of, using different technologies for each of your potential indications?
Yeah, as we think about our pipeline, so the first program we talked about, Verve 101, and targeting PCSK9 and the clinical development in the way I mentioned. The second program targets a different cholesterol-raising gene called ANGPTL3, and that's a program that it's in development right now, and expect to name a lead candidate by the end of this year for that program. And then we have prioritized additional targets, let's say programs 3, 4, and 5. And for each of these targets, we're going to take the same approach we took for the first two, which is we want to match the gene editing technology, optimal gene editing technology for the target of interest. And there's a number of considerations that go in, right? Which is efficacy, so the guide screening that Andrew mentioned, but also off-target profile and potency. And so, when we started with the first two targets, we just had a couple of technologies: standard Cas9, CRISPR-Cas9, the molecular scissors, and then we had the emerging technology of base editing, and that's what we compared. And now, in the last couple of years, there's been a proliferation of additional technologies like prime, like integrase-based approaches, and so forth, that now we should take a serious look at and try to see if any of those might be the best one for any given target. So that's the kind of evaluation that we're in the process of doing right now.
So, of course, technology is important, the modality you're using, and then we have to talk delivery, obviously, because that is sort of the next really important component. And you've done some pretty innovative things with your LMPs to, as you target the liver, to make them more susceptible, and using GalNAc as well, I believe. So would love to kind of just run through that and in terms of what you've done sort of to have a more innovative LMP.
I think that I just want to give the history, and which is back in 2018, when we started the company, Ali, we already had decided on the format for the drug. We wanted it to be mRNA, guide RNA, mRNA for the editor. The guide RNA tells the editor where to go. And then we explicitly said we want to use non-viral delivery and lipid nanoparticles. And the reason, and that back then that was a little bit of a controversial decision because the best established was virus for liver delivery. But we knew, or we thought that virus would not scale ultimately to the millions of patients we would want to reach. And viral delivery, has some drawbacks of inflammation and safety issues. And so we settled on this LMP. Now, at the time, there was no clear, LMP that would automatically work for liver delivery, particularly in non-human primates, larger animal models. And so that's kind of where Andrew stepped in 2019, and he was handed this problem of, well, look, we already know this is the format, solve the problem for us. So Andrew, what have you been able to accomplish over the last few years?
Well, exactly as Sek said, the problem was, reasonably well understood. But in the case of liver delivery, actually, there had been some progress made over the previous decade or so in liver delivery. And so what we were able to do was to build upon a lot of that work to get our lead program, Verve 101, which is a liver delivery of gene editing. I think the nuances to that is that nobody had ever really demonstrated high-level in vivo editing. I mean, they had developed these lipid nanoparticles for siRNA. They had developed them for shorter mRNA delivery. So I think what we had to do was really optimize the payloads and the interaction with the lipid nanoparticle to be able to achieve high-level in vivo editing. And that's really, I think, the bar that we crossed in 2019-20 in monkeys. The interesting challenge that emerged as we headed into 2021 and we're thinking about our second program, ANGPTL3, there's a unique patient population for that therapy. ANGPTL3, the monoclonal antibody from Regeneron, is approved now for homozygous FH. Homozygous FH is an even rare form where patients completely lack or nearly completely lack the LDL receptor in their liver. So this created a real challenge for lipid nanoparticle delivery to the liver, because that is actually the mechanism that by which lipid nanoparticles are taken up by the liver. So that drove us to really invest in lipid nanoparticle discovery for the last 3 years or so. Full spectrum, lipid nanoparticle and some targeting strategies that we were working on. You mentioned one of them, which is a GalNAc ligand that we developed that we could incorporate into lipid nanoparticles. This allowed us to get into the livers of these patients with homozygous FH because it's taken up by a different receptor, the ASGPR receptor in the liver. And so we could get taken up with high efficiency, deliver to the liver, achieve base editing at a very high efficiency in the liver in monkeys that lack the LDL receptor. We actually created a homozygous FH monkey model. I'm not sure anyone's ever done that before to try to develop a new therapy. But we created a homozygous FH monkey model. We demonstrated that our LNPs could bypass the LDL receptor and get into them. And that's really, I think an unusual example of kind of forced innovation to solve a particularly challenging problem of delivery to liver. What we've further found, though, is that in building that platform at Verve for lipid nanoparticles, we've developed a lot of lipid nanoparticles that we think are going to be useful for liver delivery. And we actually think this GalNAc LMP that we've been working on, we've shown now that it works in wild-type monkeys as well, and we think it's going to be a useful delivery tool for us for a number of our programs.
I should mention that when we got started in 2018, I think the only group that had shown high-level editing using LMP delivery for in vivo liver was Intellia Therapeutics. So they had some nice monkey data at doses of LMP that were we now know it's on the higher end, like 3 milligrams per kilogram. And so that was the bar that we had to kind of get to. And what we've been able to do over the last couple of years is get high efficiency liver editing at doses closer to 0.5 mg per kg or 1 mg per kg. So several fold more potent than what the field had previously demonstrated. So that puts us in very good shape for the first program, Verve 101. And then for the second program, as Andrew mentioned, we have this GalNAc LMP that's going to serve as the delivery vehicle.
So, all things and humans and animals aren't made equal, right? So when you think about LMP delivery and thinking about it, with mice and humans and non-human primates, we obviously see differences, and that's maybe a broader comment on how we have to do medical research. We can't go directly into humans. But we got an interesting question on Twitter, or at least I thought it was interesting. There was a paper, it looks like in Nature Nanotechnology, from the Dahlman lab, and something that I thought was interesting is they asked, there's obviously a difference when you're looking at the LMP delivery between mice, between humans, between NHPs, but what about the differences between healthy and heterozygous FH patients? Do you think that there's going to be any difference between those two groups?
Yeah, so we were concerned about that question ourselves, a few years ago, and so we've actually done a pretty extensive program to evaluate our LMPs in Verve 101 and actually also our GalNAc LMPs in models of wild-type heterozygous FH and homozygous FH, and the collective evidence from all those studies suggests that wild-type and heterozygous, their liver physiology shares a lot in common. Homozygous is really a special case. If you totally lack the LDL receptor, then lipid nanoparticles will not be taken up very well. But in the wild type and the heterozygous, you have enough LDL receptor to still take up lipid nanoparticles efficiently and deliver the RNA payload. It's important to remember about one of the nice things about base editing is that you really only need one molecule of a base editor to get into the nucleus to achieve your one base pair change. That's a little different than with Cas9, where you need to flood the cell with Cas9 and make multiple cuts often in order to achieve your therapeutic deletion or indel with Cas9. So we think that we're getting plenty of base editor into these livers in heterozygous FH.
In terms of the wild type and the LDL receptor heterozygous deficient individuals, standard LMPs are going to work just fine. It's really only an issue when you get into homozygous FH. And then, as Andrew mentioned, we feel we have a solution for that special case with the GalNAc LNP.
Having said that, we do think that the GalNAc LNPs are a nice option in that they work across the board in everybody, right? They work in wild type, they work in heterozygous, and they work in the homozygous. So, it is a nice tool to have in your toolkit in the event that we do, that we do need it.
So I don't know how much you can share on this, obviously, but would love to hear about anything else in your pipeline that you can discuss. I know a lot of times companies don't want to go too deep into their pipeline, so a lot of things are kind of held secret until they need to be not held secret. But would love to know if there's anything you can share in terms of other programs that you guys are currently working on.
Yeah, I think I'll start by just saying that, for our first two programs, we see those as kind of a pipeline in a product because of that staged expansion of addressable markets that I mentioned. So for the first program, you're going from 1 million people in the US to 22 million to, even much larger, ultimately for prevention. So that's the case for PCSK9 program, but also for ANGPTL3, you can get that same expansion starting from homozygous FH to heterozygous FH to ASCVD. So that's maybe point number one. I think in terms of beyond the first two, when we started the company, we had two other targets that we had prioritized, both liver genes, both lipoprotein genes. So that's one of those is lipoprotein little a or LPA, and then the last is a gene called ApoC3. Those two genes are still of interest to us. And the reason these four genes, PCSK9, ANGPTL3, LPA, and ApoC3 were chosen, Ali, is because they represent three distinct risk pathways, three independent risk pathways. One that's focused on LDL, and that's the PCSK9 gene. Then one focused on triglyceride-rich lipoproteins, and that's and LDL, and that's ANGPTL3. And then the third being LPA, and that's the LPA gene. So ultimately, if you had a product against each of these three, you would be able to solve much of, if not all of, atherosclerosis risk coming from cholesterol. So that's kind of where we are with cholesterol and lipoprotein genes. First two products, and then two more still kind of in research phase. Beyond these four, we have prioritized additional liver genes where manipulating that gene in the liver can lead to cardiovascular health. There are a set of genes like that, and the cardiovascular indications are maybe a bit broader than just atherosclerotic cardiovascular disease. So, for example, like cardiomyopathy or other related thrombosis. And so there is another set of genes that we are thinking through and prioritizing. And I'll probably just leave it at that.
Perfect. So some of the bears in gene editing, we'll talk about costs. At ARK, we love talking about cost declines. We typically think of, Wright's law when we talk about cost decline. So we talk about, for every cumulative doubling of units produced, costs are then going to fall by, X or a constant percentage. So, obviously we don't need to do specific calculations right now. That's what I'm for behind the scenes. But what are your thoughts on how quickly costs can potentially decline for gene editing and just the idea of these therapies becoming more and more democratized?
Yeah, this is a very important topic. And I think I'll just start with a couple of points and then let Andrew add in as well. I think when people think about gene editing or gene therapy, they're immediately thinking rare disease pricing and millions of dollars a dose. And that's not going to be our model because we ultimately want to reach millions of patients with heart attack. And so if it's not going to be millions of dollars a dose, then what is it? Well, when we started the company a few years ago, we really didn't have a great sense of could we get the cost of goods low enough to bring this to large patient populations. And but I mentioned we'd already decided on that format of mRNA, guide RNA, and LMP. Well, the last 2 years, COVID has changed this whole calculus on cost, because the COVID mRNA vaccines look just like our drug. And we know that COVID mRNA vaccines can be made very cost effectively, a couple of dollars a dose. Now, that's for 100 micrograms. We're going to be giving, let's say, milligram doses, so 35 milligrams, for example, for a 70 kilogram male or female. So, but even if you just do that extrapolation from 100 micrograms, $1 to 35 milligrams, you're still now in a very cost-effective cost of goods. And so we are very comfortable going forward that we're going to be able to make this stuff and price this in a way that is accessible to lots of people.
The only thing I'd add is I think, Ali, when a lot of people talk about your kind of bare thesis on gene editing, they're thinking about ex vivo therapies. They're thinking about cell therapies. These look very, very different from a manufacturing standpoint and a cost standpoint. So I think at the end of the day, it really comes down to, as Sek said, it's really matching your product to your target product profile. And so that was an important part of our decision making to focus on this kind of format. Was ultimately that we wanted this to be a cost-effective therapy for millions of people. And so that's been kind of front and center from the beginning of the development. And, we invest a lot of effort in on the product development side on ensuring that we can meet that vision of a cost-effective therapy.
I will say selfishly, I'm working right now on a cost decline model for gene editing. So always great to hear your perspective about that, because it's definitely a challenge figuring out all the costs that actually go into it, including just, the scientist's time. And then there's obviously manufacturing. There's obviously cost of goods. There's obviously, there's so many different costs that go into it that, are maybe not everyone thinks about. And so to get this final therapy. So it's interesting to hear your perspective on the cost for sure.
Yeah, I mean, this will ultimately end up being an infusion, our product, a one-time infusion over 60 minutes. And it's a lipid nanoparticle, with mRNA and guide RNA. And it's pretty clear that this stuff can be made at scale very cost effectively. And, we've selected indications that where the patient populations are large enough that you will need this scale. It's not a disease that affects 1,000 people. And so there's no way you can envision scale. This is a disease that affects 10% of the world. And so I think that may be a particular advantage kind of in our situation. And we have this wonderful roadmap to scale with COVID vaccines. And the manufacturing capacity for mRNA, guide RNA, and LMP the capacity is just, ballooned in the last 2 years, of course.
Yes, and mentioning that, I know we got the question all the time, what are you most excited for mRNA technologies? Vaccines, obviously, we've seen what they can do and we've seen, their potential. But, there are certain reasons why, an mRNA vaccine was particularly positive for COVID, as SARS-CoV-2 is an RNA virus. But I think one of the most interesting and potential use cases is really through gene editing. And what it's done is that so many pharmaceutical companies and others have created these, factories to produce mRNA and LMPs. And so that has really proven the cost to go down. So I think that's a really exciting premise for gene editing in general. So maybe to shift gears a little bit, I think we can never talk about gene editing without thinking about some of the hurdles that we're going to have to either go through, or some of the ones that we've already kind of gone through. And so one of them is going to be obviously regulatory. and some of those challenges that are going to come with, regulators seeing sort of a new potential class of therapies. Although, they've obviously seen it before, and you and other companies are already engaged with the FDA, but curious how you think about sort of the regulatory pathway and also some of the issues with intellectual property as well.
Yeah, I think, Ali, there's no question with a new technology, you have a certain burden to, collaborate with the agency on educating them and also understanding how they're thinking about some of the challenges and working with them. And I think that that's where we are, that's where the field is. We've had a very positive experience interacting with both the FDA and European regulators. I think at the end of the day, people recognize the unmet need. They recognize that this is a transformative therapy. And so the question is really, how do we collectively work to ensure that there's really high quality science behind them and that we understand the mechanism and we understand all of the potential safety concerns on the CMC side, on the off-target side, on the toxicology side. And that's really what we've done. I wouldn't say there's any single, kind of magic bullet to that approach. It's really just being, I think, open-minded and collaborative and working through all of the questions that agencies have, and addressing them.
Andrew, do you want to walk Ali through some of those IND enabling studies that we've done to kind of that'll be going into the package
Sure.
for the regulatory submissions this year?
Yeah. So, there's two key aspects of the GLP tox package. One is our extensive non-human primate data and a GLP extension of that, so demonstrating safety in non-human primate. The second is a similar toxicology study in a mouse disease model, so in heterozygous FH disease model. So those are the two core portions of the tox package. Off-target is a critical area for us. We've been very transparent with the agencies and actually with the public as well about a lot of the off-target data we've been generating at Verve, which has shown that we have exquisite specificity. So, when we look at the single site that we, in PCSK9 that we want to edit, we see very high efficiency editing, and it's a very predictable edit. So we don't see a lot of unpredictable changes at the on-target site. And then the next step is to look at all the other sites in the genome where editing might occur. So we've developed panels of more than 3,000 such potential off-target sites around the genome. And these are sites that maybe look a little bit like our on-target site in PCSK9, or for some other reason might be at increased risk of off-target editing. And we look at all those sites, and we've shown that we do not see off-target editing at any of those sites in the liver. So we've gone through and built a very substantial package on the off-target side for regulators, addressing each of the questions around guide-dependent editing, guide-independent editing, as well as the propensity to develop structural variants that have been seen with other nucleases like Cas9.
The other thing I should add is that, Ali, when you do an infusion into the bloodstream of the drug is going to go to the liver. Almost all of it goes to the liver, but it is systemically infused. So there's a possibility the drug could go to other organs, and there could be editing happening in other organs, off-target editing. So here, it's been a very productive discussion with the FDA, where they guided us to evaluate off-target editing, evaluate for off-target editing in tissues where there might be biodistribution of the drug. So first, check the biodistribution, and then second, evaluate for off-target editing potentially in those other tissues. And that's exactly what we've done. We've shown, we've released data on the biodistribution of our drug when it's infused into monkeys, and it goes to liver, but also two other tissues, the spleen and the adrenal gland. And then our off-target analyses have now been in liver cells treated with drug, but also in primary human splenic cells treated with drug, as well as primary adrenal cells treated with drug. And so that's another facet of off-target analysis in vivo for in vivo liver health.
Obviously, we cannot have a podcast without discussing off-target editing. I don't think it will ever happen. And obviously, that's one of the largest challenges. I think, just uncertainty and nervousness around it. And I think, going back to something you said at the beginning of the podcast, these are non-viral delivery vectors, and so a lot of the sort of off-target edits or sort of safety challenges that we've heard of from others has been more focused on viral delivery. So it's important just to make that distinction, I think. And I don't know if you wanted to add anything to that, Saif or Andrew.
I think that, as I mentioned earlier, we made an explicit decision to move away from virus, mainly for some of the reasons that we've talked about, which is the inflammatory reaction, the liver function tests that can rise and then stay elevated over time. And then the durability. Actually, this might be a good time to talk about durability, because that's another reason, another particular advantage, I think, of gene editing versus, let's say, viral gene therapy, where there have been some issues around durability based on immune reactions, for example, that take away the protein that's being expressed or the vector that's being expressed. So the reason that gene editing is likely to be durable for the lifetime of the animal, at least based on the emerging data, is that when you go in and make the edit, let's say in liver cells, that edit is being generated in mature hepatocytes. And the liver, for example, does turn over. So every couple of 100 days in monkeys and humans, you have a whole new set of liver cells than what you had before. So the entire liver turns over. Now, what is the source of that turnover? It turns out the source of that turnover are mature hepatocytes. So when we come in with our therapy, for example, and edit at time zero, you're editing all the mature hepatocytes. As I mentioned, we have like 100% efficiency, like all the hepatocytes. So when those hepatocytes divide to give rise to the next generation of hepatocytes, the edit is carried forward. So that's why there's likely to be durability for the lifetime of the animal. Now, what evidence do we have for that? Well, there's been data by Intellia, for example, with monkeys out to 2 to 3 years after editing. The editing is maintained. We have data out to almost a year and a half, 18 months. And then in some forced regeneration experiments that we and others have done in mice, there's a very strong durability. So I think this whole durability question, the answer is going to be very different for gene editing as compared to gene therapy.
Especially gene editing in the liver, because of that turnover features that Saif just walked through. That may not, we may not see the same level of durability in other tissues because stem cells may play a different role in those in turnover of those other tissues. But in the liver, we're pretty confident this is going to be a lifetime therapy.
Yeah, that's really good context. I think a lot of people forget that gene therapy and gene editing are not the same thing. So that's really good context for people. So thanks for walking us through that. And obviously, I think durability on all of these modalities is still, somewhat of a question, but thanks for walking us through sort of how you guys think about it and the possibilities for that specific therapy, which is pretty impressive and interesting, and we'll certainly be watching. But I wanted to pivot a little bit just because this is ARK and our focus is obviously on disruptive innovation. So wanted to ask if you had any thoughts or if you guys are implementing anything to sort of help with this, which is how do you further reduce time to market for new therapies? So, what do you think it will be that will help us get to market even quicker? At ARK, we focused on things like better sequencing technology, neural network-based algorithms, like AlphaFold that can predict protein folding, or things like CRISPR even. So we published a lot about this in our Big Ideas deck, actually from 2020, just on the intersection of AI, CRISPR, next generation sequencing, and how that really can improve time to market and also trial phase failure reductions. We estimate by 25% each. But I'd love to hear your thoughts on if Verve is doing anything to get to the clinic faster, reduce failure rates, and if not, sort of how you think Verve can, further improve and add new modalities or new technology to the mix in order to help with that.
Yeah, I think our secret sauce, I think, is a lot less sophisticated than AI, and it's just focus. We started in 2018 with a laser-like focus on a couple of targets. We knew what we wanted to do and which organ we wanted to do it in. We had that format, and then we had to bring all the pieces together. And where there were no pieces, we had to, in a couple of cases, invent or discover ourselves. And the entire team, now of 130 people at Verve, is focused on getting the first drug to patients. And we fully expect this year to dose our first patient in the second half of this year. So it's really like 3.5 years for a brand new treatment modality. A lot has gone into this, but I would say one of the biggest drivers has been that focus on product development.
Yeah, I think I'll kind of look to the future, Ali, and into clinical development. I think a few things. One, we're targeting a gene and a pathway that's very well understood with excellent biomarkers. So as we go through our clinical development, we're not flying blind. We know exactly how much PCSK9 reduction we need to achieve in our patients to achieve a clinical benefit. So that's going to allow us to move much more quickly from phase 1 to phase 2 and 3 than, say, a therapy where the amount of editing that you need to achieve a therapeutic effect is unknown, or where you can't monitor that very easily. So we have by virtue of the kind of careful thought that's gone into the target, I think that's going to let us move with a higher degree of confidence and a higher probability of success through phase 1 and into phase 2. And then thinking about phase 3, a couple important points that I think are going to be really helpful to us in terms of getting regulatory approval quickly and getting to market. One, our clinical endpoint is LDL lowering. So this is the clinical endpoint for heterozygous FH patient. Every drug approved for heterozygous FH has been approved based on LDL lowering endpoint. And that's an endpoint that you can hit that endpoint with 80 patients. You don't need large numbers of patients to hit that endpoint. Now, of course, our clinical development will be with many more patients than that, but that's to build the safety database. Our efficacy endpoints are very high probability endpoints and very well understood and very well characterized. So I think there's a number of aspects of our clinical development that we think are going to make it for very efficient clinical development. And then on the operational side, I think we can talk about this more in the future, but we certainly are implementing a lot of the innovations in clinical operational efficiencies around remote monitoring and hybrid structure, particularly as we look to some of the long-term follow-up that we're going to be doing. So we're going to be dosing people once and then following them to look at durability as well as safety. And so we're looking at ways we can do that very efficiently in a modern fashion that is enabled by technology.
And also even just in the lab. I've been lucky enough to visit it, and I think I would say that it's very impressive in terms of the amount of technology you have in the lab, in terms of automation, being able to monitor certain things at home instead of running back to the lab, robotics. So I think there's a lot of automation that goes in there, which is very helpful for sort of the I guess, the preclinical aspect. So I think that's really sort of one of the unique characterizations that helps with reducing costs eventually, as they're expensive at the beginning, but then also helps with getting things to clinic quicker, which then helps with commercialization, of course. So a funny question. I opened this up to Twitter and I asked for questions on Twitter, and I actually knew this question would come up and was thinking about it myself. But the question was, Sek, you're very active on Twitter, and you engage a lot on Twitter. And I think the question was something about, do you think that this is a really interesting and important way for you to engage with the community? Andrew is also active on Twitter, by the way, so just want to highlight that.
150th the followers. So.
But maybe not after this, Andrew. So you never know. But I think the question was, like, do you find this a really interesting way to kind of interact with the community at large? And I think someone even referenced kind of like, is this helpful from an IR perspective? And I know ARK is certainly active on Twitter as well, and I find it really helpful when people make comments or link articles. So just curious on your thoughts sort of as in the Twitterverse.
Yeah, I mean, I love Twitter. I think I've been on. It sounds like I think it's my 10th. It's like the 10th anniversary of kind of being on Twitter this year. So I started in 2012, and I learned something every day, multiple things. And I think the key to to making effective use of Twitter is curating your feed, avoiding distractions like politics, for example. And really staying laser focused on delivering info. I find that what people want from me is new information. And so when I was an academic, I enjoyed kind of talking about the research we were doing, the new findings, trying to explain it to a general audience. And then having made the transition now to the company, I'm giving people kind of a window into what it is like to build something from the ground up. In terms of having a certain focus, a certain mission, bringing the team together, and then, we're executing. And so, and that execution involves the science, but it's also some of the business stuff. We went through the IPO, we went, raised capital and all those things. And even some of the day-to-day stuff, like, we've had to, do all of this work over the last 2 years, 2.5 years during the pandemic. And very proud of the fact that, we've had our employees, come in, the lab-based employees, the whole pandemic. And we've been doing surveillance testing 2 to 3 times a week the whole time. And I put out some information recently on our testing results, and I thought this was a nice window into kind of how, a biotech in Boston has experienced the pandemic. We did our testing from July 1, 2020 is when we started. And then to December 31st of last year, so 18 months, we had done 4,300 tests, PCR tests, surveillance tests, and we had 3 positives of the 4,300. So really very reflective, I think, of how great our employees take care of themselves in terms of, their behaviors. And so that was great. And then, but it was something we put together to, make the workplace safer. And then we had January, the first week with Omicron, and we had the first 140 tests the first week in January. We had 14 positives. And then the second week was 4. The third week was 1. And then this last week was 0. So, we experienced what, the rest of the world is experiencing. But anyway, getting back to Twitter, sharing all of this, people love new information and they respond to it. And then lastly, I would say is I love engaging. You have to pick the people to engage with. There's a lot of noise in the system, but when you do pick the right people to engage with, you learn, they learn, and there can be very mutual beneficial exchange.
Andrew, nothing to add on Twitter.
No, I think Sek is the master, so I have nothing to add.
Got it. Yeah, I mean, I totally agree. I think having a curated feed and then just, learning from each other is really amazing. I know I was thinking of the posts that have stuck out for me, but I know like even when a journal article comes out or if, Verve has put something out that's new, it's so easy to go on Twitter and see that you've already posted about it. And not only have you posted about it, but you typically then like explain what that article is saying so that, if someone doesn't have a scientific background, they may be able to digest it a little bit better. So I think it's helpful, but everyone has their own, perspective on social media. But I definitely thank you for it. I also remember I was thinking of the post that stuck out the most, and it was probably I think it was like your Thanksgiving. I think you had posted like a picture of all the food. So I don't know why. Maybe I was hungry that day.
Well, that's actually one of the other lessons besides people crave information, and they're usually following you for a very specific thing. So if you make sure whatever your posts are, like 80% of my posts are about the company, LDL, atherosclerotic cardiovascular disease, but then they also like to see a little bit of the personal side, so I do, post things like dosa and, things that I like from my background, food. But the second thing people like is we're a very visual species. So people like images. And that's an important lesson. And Andrew is amazing at actually developing images for our data, for slides. And it's really critical that you are able to convey information not through text, but actually through images. And that's one of the things I learned in the 10 years on Twitter.
Andrew, I'm now curious, how have you become a master at developing images? Are these PowerPoint images that you're talking about or data slides?
Well, it's data representation. It's communicating science in a way that really gets at the key message that people are looking to understand from the data. Often people have a lot of data and it's kind of just, there's extraneous stuff on the slide. A lot of it's just being very rigorous about editing yourself down.
I'm going to embarrass Andrew, but he is amazing at putting pen to paper and just creating beautiful representations of the data generated by the group.
Well, when you're building a company, one of the things that you learn is you have to change your role over time. And, but at the early days, and honestly, until fairly recently, Sek and I, made the vast majority of the slides for Verve ourselves. And, we're building a team now. And so we have, building strength across the organization. But you really do have to be somebody who is, you gotta do it yourself sometimes.
So I definitely am always working on data visualization. So now I know Andrew, watch out. Because if I ever have questions now, I'm going to be like, all right, well, found the master. So that's a good tidbit to know. And obviously Verve's slides are always really digestible and good to understand. So now we know the master behind it. Another question on Twitter actually also was about any funny or early stories at Verve. So I guess we kind of opened that Pandora's box. So not sure if there's anything else you wanted to share or highlight there, but
I don't know if there's any early funny stories, Andrew.
Well, we always joke with our the other Andrew, the third portion of our early management team. We've of course grown it recently with the addition of Allison Dorval, our CFO. But in the spirit of everybody having to do everything, we famously have moved as a company like 4 times in 4 years because we keep outgrowing our space. And Andrew keeps telling us, we just need this space, and Sek and I keep looking at it going, bigger. But famously, when we moved into one of our first spaces, Andrew,
it was actually in 26 Lansdowne Street, where we sublet it from Beam.
Right. Yeah. We had to. Our COO was there all night assembling all of our chairs and all of our desks for the company because that's what you do when you're building a company. So.
Wow, that's amazing. Okay, well, I'll give you the opportunity. Have I missed anything? Is there anything you want to highlight about Verve before we sign off?
No, I just highlight that. Look, it's all about solving this disease, and the solution. When I look back, when we started, it was very clear. We had conviction that the solution to this problem is a one and done to lower LDL, and it's just been very satisfying to develop that product over a relatively short period of time. And then this is the key year for the company. We're really going to be able to take this to patients with disease and hopefully prove efficacy and then expand from there. So really amazing journey, and very grateful to Andrew and the rest of the team to be able to do this. And thanks for talking to us.
Of course. Well, thanks so much, you guys, for joining. I know we'll hear a lot more from you guys on Twitter, at conferences. And so, yeah, thanks for joining and for the work that you're doing, and hope to talk to you guys soon.
Thank you, Ali.
Thank you, Ali.
ARK believes that the information presented is accurate and was obtained from sources that ARK believes to be reliable. However, ARK does not guarantee the accuracy or completeness of any information, and such information may be subject to change without notice from ARK. Historical results are not indications of future results. Certain of the statements contained in this podcast may be statements of future expectations and other forward-looking statements that are based on ARK's current views and assumptions, and involve known and unknown risks and uncertainties that could cause actual results, performance, or events to differ materially from those expressed or implied in such statements.